Do you want me to
Okay.
do you want me to do my thing?
What do you mean by your thing?
Hello and welcome to the second episode of No Filter Fauna.
I love it. It's perfect. Juve, I have three things to tell you.
Yes. Okay.
Number one, I just saw on TikTok that today, 1 year ago, is when I posted the Naked Mole Rat video that you made. How perfect, timing wise, is
How
this?
perfect.
Here we are recording today.
Amazing.
Thing number two.
Okay.
I have a friend on TikTok who I don't know him in real life, but we know the same people because when you know one manatee person, you know all the manatee people.
Right.
His handle on TikTok is Dr. Seaweed.
Oh, he now follows me, even though my TikTok is very
Yay!
boring.
Okay.
Empty.
So he is, it will come as no surprise, he studies macroalgae.
Amazing.
He said that they use the term spatulate all the time.
That is a throwback from our first
Because
episode,
it's a leaf
audience.
thing. Right. So yeah, so if you haven't listened to the first episode, definitely listen to that.
Amazing.
And just enjoy our reactions to the term spatulate.
Oh my gosh.
So yeah, he was like, "Oh, that's so funny. This is a word we use all the time." I'm like,
what do they
it.
use it for? That's the important thing.
Describing leaves.
Oh,
because
okay.
it's leaf shape. Yeah.
Okay, that makes sense. Yeah. Once you told me the word and like in this, I was like, oh yeah, I have seen that in botany books, like tree ID books, because you're looking at the leaves and it's like, is it serrated? Is it spatulate? Is it lobed?
Oh, I don't think I've ever read a botany book.
What were we up to?
Number 3.
Okay, 3.
I mentioned in the last episode
Okay.
that I have been told all my life to like be quiet, and that people tell me I'm yelling, my husband especially, and like, if you want me to definitely be louder, tell me to quiet down.
Right,
for
obviously.
sure.
Yeah.
And like, I'm an excitable person, I'm the excitable scientist. After like watching and listening through our whole episode, for the first time in my life, I understand what people are talking about.
I feel like the New England coastal kin. There's a kin. Yeah, there's a kinship there. We're all just fucking loud. It was
so loud.
So loud.
I am moving back to New England in October from Florida. And I am so excited.
We are so excited to have you back up here.
Thank you. My friend said something yesterday about making acorn cap whistles. And I haven't seen an acorn in 7 long years. The oak trees here don't have them. They're different oak trees. And so,
sweet little baby.
Like, they're just those little things. And I'm like, oh, man, acorns.
Acorns. Whole on
acorns. And just our woods. I like the woods of New England. As humid as Florida is, the forests feel drier. And so it doesn't feel like there's just not as much leafiness. It's also, there's a big lack of deciduous trees. It's mostly conifers in our forest. It's mostly like sand pines and different kinds of pines, plus the live oaks.
Got you.
So I miss like maple saplings,
just little babies, the little babies.
Just little babies.
And little sassafras. Sassafras is what I love about up here. I don't know if it's anywhere else, but I love the sassafras up here.
Probably. But yeah, I don't know.
I don't know the range.
I don't know if it's down here.
Yeah. You can find the range, I guess.
But I definitely loved as a kid, like picking it and like peeling the bark and licking it.
Licking it. Listeners, if you didn't know, sassafras, if you crumple most parts of it, I believe, it smells like Fruit Loops.
It does not smell like Fruit Loops to me.
What is it? No, we can't have the 2 episodes in a row. Damn it.
I get like a root beer or birch beer.
Okay, no, that is what people say it smells like too. Like, I'm not. Okay. To me, it smells like Fruit Loop. That is how it was introduced to me, as this is the Fruit Loop smelling tree.
Okay, interesting.
So that is, but I can definitely see that it has that scent as well.
Yes. Okay. Oh yeah, we got to talk about
we have guests.
Our guests.
We have guests, and
I'm
they're
so
coming
excited.
soon. So I guess oh my goodness. So, listeners, if you listened in on episode 1, or if you know anything about the Excitable Scientist on TikTok, Amber Lea and I both went to the same graduate school, and that graduate school was the Masters of Animal Behavior and Conservation program at Hunter College in New York City.
Oh, you know what? Hold, please.
Thank you for that. Um,
Jesus
the
Christ.
most New York thing.
And here you are. You've been trying for an hour to introduce Dan and Antonella.
But we're gonna do it.
But I had to play a DJ horn.
So this will get personal for me. So, yeah. So my master's thesis was the target species of that was naked mole rats. And on your TikTok, Excitable Scientist, I apparently became very popular over my post on naked mole rats. But there is a greater authority, and that authority is my mentor and thesis advisor, Dr. Daniel McCloskey. He's gonna die. I'm gonna send him a clip.
Why do I do this?
Anyways, so Dr. Dan McCloskey is an associate professor at the College of Staten Island, and he has been researching naked mole rats for, gosh, he'll probably say how long it's been, but I think it's been at least since 2010.
Okay.
For quite a long while.
It's a good while, yeah.
It is. And he is an awesome human. He has won an award from President Obama for his cool scienciness. I know we are going to be in the presence of greatness.
Did he, when he won the award, did he get to meet President Obama?
Yes. There's a picture of him at the White House and everything. I know. I know.
That's amazing.
It's really, the man is amazing. He has done so many great things for science, for naked mole rat research. He is just a good person all around, and so I'm really excited to have him on to talk about his favorite, parts of naked mole rat research, what he does, and along with him, we're going to be talking to his PhD student, Antonella, and she is going to describe her research as well, which I'm very excited for.
That's great, yeah.
Yeah.
Is she at the College of Staten Island?
Yes.
Okay.
Yes, and so for anyone who doesn't know, once you're within the City University of New York, CUNY system, you can generally study with anyone in the CUNY system, and it depends on the program. So for our program at Hunter, I needed a faculty member that was full-time at Hunter to also be a second advisor, while he was my first.
Who was your other advisor?
My other advisor was Dr. Diana Reese, who was the program director, and is a very famous dolphin researcher, cognition, right? Cognition research.
Yeah, dolphin cognition behavior.
World renowned, and a great lady. We adore her. Maybe one day she'll agree to be on the show.
I was just gonna say, I bet we could get her on the show.
I think she might enjoy it.
I bet we could.
Anyway, so I did my master's with Dan. I'm just gonna call him Dan. I can't pull the Dr. McCloskey. I'm just letting everyone know now. Because he's Dan the man. We call him Dan the man.
That's awesome. I think it's indicative of the relationship that you have and the kind of person that an advisor is when you call them by their first name, or at least not Dr. My advisor was Dr. Peter Moeller. I call him Peter in email. To his face, I've never called him anything other than Moeller. Like, when I talk about him, we talk about Moeller. It was how everybody discussed him in our program. It was always just Moeller. And, like, he would never be the kind of person who's like, please, it's Dr. Moeller.
Yeah, there are some people that are like that. Yeah.
Yeah, it's nice to find the ones that are
right.
Just, I'm a human, you're a human. Call me by my name.
And Dan is and you will see he's absolutely like that for all of the greatness that he is and things that he has done. Still a very humble human, and we love that. Good vibes here.
Love that. Yes.
Yeah, and so I actually, so it's a sort of convoluted thing. So I went to the College of Staten Island for my undergraduate, which was biology. And I actually had class with Dan when I was like 19 or 20.
Nice.
And I was working as a zookeeper at that time. And then when I left the zoo, I came to the College of Staten Island to work as a laboratory animal technician. And that's where Dan's animals are. And so I got to take care of Dan's animals, his naked mole rats. And as I went into grad school and started thinking about what am I going to do? What am I going to do? It kind of just happened that Dan was looking for someone to do a project to help improve the layout and the enclosure type that we have for the naked mole rats. And it's funny because he kept on giving that project to undergrads. And as I dove into it, it was like, this is not an undergraduate project. Like, this is very involved.
Yeah.
And so here we are now, like 6 years later, and I did my master's. We did a project, and we're still working out the kinks of it. Hopefully, we'll get it published soon. Because listeners, publishing takes a while.
Oh my god, it's gonna be really hard.
Takes a long while,
especially when one or more team members has like ADHD and things kind of languish.
Mm-hmm. Or a new job
or life,
and they're trying to settle in, and it's a lot.
Yeah.
And so so Dan's general shtick is everything about naked mole rats, and naked mole rats are really cool, and I'll have him go into it because you don't need me talking about it.
I think first,
please,
let's pump the brakes on the imposter syndrome. You know a lot about naked mole rats.
I do, but
and I think you and Dan will complement each other, and Antonella.
Oh yes.
And I have lots of questions prepared about them, and I'm so excited to learn about them because I know a little bit about them, but literally everything I know about them is from you. And so, like, we haven't sat for 10 hours to discuss them, so I think there's a lot for me to learn still, which I'm really excited about.
Yeah, absolutely.
I also want to mention for our listeners and viewers that I think it's an important thing to bring up that regardless of your feeling on laboratory animals, because I think there's a big spectrum of feelings on laboratory animals, one thing that I want people to know that I think a lot of people don't know is that, especially now, now that we're in like the 2020s, there are a lot of standards and principles set forth to ensure that lab animals are taken care of well, both physically and mentally.
Thank you.
Which has not always been the case. And so I think when people hear lab animal enrichment, I want to kind of point out that regardless of your feelings on lab animals, their conditions and care have gotten a lot better.
So much better
over time. And so there are people like you who are, your purpose, your job is to continue that and continue to make things better for lab animals. And I have mixed feelings about it because I'm like, I feel bad for animals that are like tested on and whatever. I'm 100% against things like makeup and like cosmetic things that are not
it generally
for the purpose of human health.
Yeah, it needs to be very well justified in general.
But I think it's nice to know that they're the ones that are really, we'll learn in the case of naked mole rats, that are really making huge strides in helping scientists make discoveries to make human lives better and animal lives better, are really well taken care of. And so I think a lot of people don't know that, and it's at least makes me feel better knowing that.
Yeah, and I used to in the facility that I worked with with Dan, we would bring in classes and stuff, and I would say, first of all, there is federal regulations that you have to, every scientist needs, and you've been through this, every scientist needs to write an IACUC approved protocol. And that process, you need to justify why you need to use that animal, how you're going to use that animal. Is that animal going to experience pain, surgical procedures, what type? And the biggest thing, at least for me in my eyes, is what is the minimum number of animals that you need to use to produce a significant result? And now, I'm not saying statistically significant, because there are different types of studies that don't always rely on that. But, especially for like clinical trials, it'll be statistical significance. But it could also be, especially with behavior, what is the significance and how are you going to show its significance and its effect size and blah, blah, blah.
Right.
And so I do say to people, anyone who's uncomfortable, if you are uncomfortable with it and you want to do something to, change the field, what I suggest is definitely write to your representatives. And any research that's being done, you ask for more specific regulations to be had. Because generally what we use in the laboratory animal field is guidelines. And, some of them are like hard and fast requirements that your animals need X, Y, and Z, should have enough space to do whatever, should be able to turn around, to stand up and stuff. But get extra specific. Make those minimums even better and all encompassing. That's what, incorporate the best knowledge that we have available to us about their wild counterparts, about, their behavior, their psychological well-being, anything like that. That's where the change will, almost overnight, that
Oh,
change will
wow.
be immense. Yeah. I mean, there's legislation, like loopholes and stuff that, like, the biggest thing is, animals that are considered animals, or animals that are regulated species. So like for USDA, mice and rats and things that are bred for research aren't really considered regulated species, and therefore kind of fall outside of being an animal in a weird way. It's a weird thing. And because of that, there's guidelines to treat them a certain way. Um, but there's not necessarily like these hard and fasts, when it concerns like enrichment and behavior. There are other regulations and federal regulatory statutes that, like, dictate what you should have and what you shouldn't have. And, in general, mice, rats who have been used in laboratories for almost 200 years, it has been, a great increase in how we take care of them and stuff.
Yeah. Oh,
Um,
absolutely.
but this would be something that overnight would really be effective and productive. So,
Yeah. And I think it's nice for people to have something that they feel like they can actually affect real change, because
yeah,
I think
yeah,
a lot of times we wonder if we get listened to if we, send an email to our representatives or something. But I think they do pay attention to this kind of stuff.
yeah,
And if you're completely 100% against laboratory animals, that's also very valid. And you can
absolutely.
write that, and I
Yeah.
think it's, you know,
Any
everybody from
pressure
different.
is going to net a positive, I
Yeah,
feel. And,
I agree.
and most everyone who's ever worked in the laboratory animal field is always like, yeah, the day that we don't have to do our job is a really good day.
Mm-hmm.
Um,
Mm-hmm.
So when we can find those
Which
alternatives, which,
I want to make a small point
yeah,
that I think is something to think about for
yeah,
some
please.
people.
Yeah,
This is one arena in which I think AI can be really helpful, because I
I believe
think
so too.
I think there will be, we've already, there's a lot of science going on with computer modeling, so
Yeah,
that you can I don't understand it enough to like fully explain how it works, but basically you can write program that will show you how, like, a rat's body would react to a certain type of
yes,
medication given what's in the
yes.
medication. And then I think AI is going to help us take it a step further.
Mm-hmm.
I think there's a lot of fear around the thought of AI being used in like medication development.
Yes.
But the thing is, it's not that AI is going to
It's a
like
tool. It's a
make a
tool.
medication and
Yeah.
give it to people. It's that you can use AI to more quickly and with fewer resources model what the outcome would be of a certain
Right.
scenario.
Right.
And I'm personally very excited about
Yeah. Well,
that.
I mean, that's like, everything needs to be done ethically, and AI has not been off to a great start in a lot of ways with that.
No.
But the tool is here, and it
Yeah.
can help this issue, at least in a lot
Mm-hmm.
of ways. And so I'm hopeful for that too, because it's
Mm-hmm.
already the possibility is there. Someone just needs to take it and do it well and
Yeah.
not pull a
And
Jurassic Park.
ethically. And
Yeah.
yeah. Oh my God. Yeah.
Let's not pull a Jurassic Park,
Yeah.
please, people.
There are certain lessons that I think we can learn from works of fiction.
Oh, there are so many lessons, so many good lessons.
Mm-hmm. Mm-hmm.
Yeah, so to anyone who feels uncomfortable with what we're about to talk about, understandable.
Mm-hmm.
Completely understandable, completely valid. We would not have the Animal Welfare Act, which is the first piece of really good legislature, without people who said this is not okay.
Yeah.
And so I am never fully against anybody being against any of
Yeah.
that, even
Yeah.
with zoos. Like, put
Yeah.
pressure in places, make sure that if we're going to do these things, they're done to the best of our goddamn abilities. That's,
Yeah.
um,
Absolutely.
yeah. Welcome, both of you, to No Filter Fauna, where we've got no filter and we talk about fauna.
Great.
That's right.
So,
Um,
Amber Lea, take it away.
I I have question. I have my own questions. I have questions I've been assigned to ask by my friends. But Dan, can you start by giving us an overview of like what is cool about naked mole rats that makes them a researchable animal for you?
Okay. I knew less about mole rats probably than most people. And I'm embarrassed to say probably how old I was when I first heard the word naked mole rat. I think I was probably 35. Had no idea what the species was. And my sister-in-law, who gave me a Christmas present, which was a Fact a Day calendar in 2007 for the 2008 year. She gave me this calendar, and each day I would rip off the page and learn a new fact. And on April 17th, 2008, I learned that there was something called an African naked mole rat, and that, according to this calendar, was a cold blooded mammal. And I thought to myself, wow, that's cool. But that is probably the least cool thing about naked mole rats. So, but it was cool for me at the time because we wanted to understand how temperature affected the brain, and having a mammal with a brain like ours that we could change the temperature of was really powerful. And that was sort of the first thing that got me thinking about naked mole rats as a topic, somebody to study, to see the effects directly of temperature on how the brain performs. And like I said, I started to learn as much as I could about them and learned how really cool they were. And so the real cool thing about them is that they're really the humans of rodents, right? So they're they're less hairy than all of their relatives. They're much more social, live longer. So they have a lot in common with us compared to our relatives, right? So our primate relatives are more hairy, they live shorter, and they're less social than we are. So by studying naked mole rats, we have a friend, we have an ally that we can kind of point to and see how they negotiate their social world, right? They have a queen, so they're what we call eusocial. They have a queen and a breeding male that does all of the breeding for a colony, and all of the other members of the colony are basically brothers and sisters over many, many generations. So because they live so long, we know that they can live 30 plus years in captivity. You can have potentially 30-year-old brothers and sisters to newborn babies. And the brothers and sisters help to maintain the colony, help to raise the offspring. And so you have this whole little city of animals that live together in their own little society, and they make it work. They figure out how they're going to do everything, and they're really fascinating to study. So I sort of backed into them in a way as a research subject. I wasn't, I wish I could say, yeah, it was just this grand design that I had planned it all out, but it wasn't like that at all. It's just sort of, I learned one cool thing about them, and it happened to be the least cool thing about them, but learned so much more as time went on.
I mean, that's mostly how most research
I was just going to say, I feel like that's the best way to get started in an animal.
It's the best research. Yeah, the best research you sort of back your way into. Yeah.
Just fall into it.
Yeah. And then Antonella, how did you get involved in Dan's lab?
Oy. This might be half of a confession, maybe some information Dan doesn't even know.
Oh, okay.
This might come out.
Big reveal. I like it.
Yeah, big reveal. On a personal note, Dan was sort of somehow like mentoring me distally, right? Whenever I needed something, I would sort of go to Dan. I was checking in maybe annually, right? He wasn't a direct mentor at the time. I was actually looking to do dog research, which and then the same thing, right? I stumbled upon these naked mole rats, right? By getting to know Dan. And believe it or not, I'm not, not interested in dogs anymore. I'm getting to see the relationship or the light that naked mole rats can shine on our relationships with dogs. So there's an interaction there.
Can you elaborate on that a little bit? Like what are some things? And my dog is like walking over as we speak.
I want everyone to know this is like my first time hearing Antonella talk about this.
Oh, really?
Really?
Oh,
Yeah.
Really? Oh,
yeah.
Okay. So my brain, Amber Lea, just by default makes everything about dogs. Just by default.
Perfect. I love this.
So as I began to study naked mole rats, I was fortunate enough to, be able to work in Dan's lab. I started to see that just the way humans have co-evolved, dogs and humans co-evolve, right? Or have co-evolved. And there's this mutualistic relationship. Perhaps so too between subcastes of mole rats. So the groups within the larger group, there's the coevolution and a mutualistic relationship that they depend on, right?
So they're not all like treating each other the same.
Dan can answer that better, but so as I'm learning, all right, I think the answer is no, right? So there are groups within the group and they depend on one another, right? And so then in my mind, I have the analogy, right, between dogs and humans. And I think that naked mole rats can teach us about that codependency between dogs, between humans and humans even, right? Subgroups of humans. They're fascinating on the physiological level, on the behavioral level, right? They can open our eyes to so many things. Yeah.
So for listeners, I guess we should catch them up to speed. So the things that make naked mole rats cool are they're cold-blooded, which is poikilothermal.
Yeah, they're not really cold-blooded. They're close though. The fact today calendar was a little bit off, but that's okay.
I enjoy saying that they're just really bad at being mammals.
Yeah.
Like that's how I usually get it across. So they are very highly resistant to cancer.
Correct.
They can survive in very low percentage oxygen for very long amounts of time. Anything else that I'm missing?
Live a really long time. And their social structure is really, for mammals, there's two species that we know of that are what we would consider eusocial mammals that have a queen: African naked mole rats and Damaraland mole rats. So there's two species that clearly live like bees, ants, termites. And what's cool about them is that there's this guy Richard Alexander, who in the 70s, so our mole rats came over, as far as we know, with Richard Alexander. We think we were told in his pockets on an airline in the 70s. Yeah, like pre-TSA for sure, right? So yeah, he stuffed some in his pockets, maybe a breeding pair or something, and that's a long plane ride. And I think I'm comfortable with these animals. I am not that comfortable. I couldn't sit with them in my pockets for a 12-hour flight, especially, planes in the 70s with cigarette smoke and everything else, right?
I can't fly without falling asleep. I would fall asleep, they would be gone. I'd wake up, no clue where my mole rats were.
Before snakes maybe that's why they put the snakes on the plane, because there was mole rats on the plane first. So anyway, he brought them back. But before he went to see them, he made this prediction in the early 70s, late 60s to say, I bet that there is a mammal, a rodent who lives underground, who lives with a queen and a breeding male, just like bees, termites, and ants. I mean, how smart can you be to predict a species, and everybody's like, no, couldn't be.
Had he already secretly discovered naked mole rats and was trying to make himself look smart?
No, because he was told, oh, you better go talk to Jennifer Jarvis, who was really the queen, the queen of the mole rat universe, and she was in Africa and doing some early research, just the first one to say that they were actually eusocial. And he went to visit her, and that's where he came back with the mole rats in his pockets. But he had no idea that they would exist. But really, he was an evolutionary biologist and said evolution, if this doesn't exist now, it will one day, because evolution kind of demands it because of selection. So it was really, really cool.
An evolutionary Nostradamus.
Yeah, exactly. That's a great way to put it.
That's crazy.
Are all the naked mole rats in the U.S. now from that lineage, or have subsequent lineages been brought over?
We know subsequent lineages have been brought over. There's a really great researcher named Colleen Ingram who she's gone and collected some tissue from different mole rat groups in the U.S. and compares the genetics and can tell us that they came from different lineages. She can identify which ones came from which part of East Africa. And through her research with genetics, she found out something really cool. So we in my mind, and I think Antonella's mind, I think we haven't even talked about the coolest part of naked mole rats yet. So
which
we'll get there. So what Colleen Ingram shows with her data is that not all mole rat colonies are related, right? So it's not just about this constant outbreeding, I guess, where, maybe a pair runs off and starts a new colony. And so they're all not from the same lineage, just spreading out over time, but there are different families that date back millions of years, right? And so the striking thing for her was that you can find relatives across rivers or over mountain ranges where there was simply no way that they could have dug their way there, right? So she shows that it's sort of required that some naked mole rats have gone above ground. And the cool part that I like to think about, and me and Antonella talk constantly about, are what we call the dispersers, the naked mole rats who run away. So most of them are quite content to spend their whole 30-plus year life. It's much shorter in the wild, but in captivity, spend their whole 30-plus year life with mom and dad and all their brothers and sisters. But every once in a while, there's an animal that runs away and presumably starts its own colony, but we know nothing about how this really goes down. Are they bumping into another solo animal in the middle of the night? Are they digging down into an existing colony? So it's sort of this big mystery. When did these animals start, these dispersers? Where do they come from? How do they meet up? How do they start new colonies? So there's a lot of mystery there about really the initial reproductive activity of naked mole rats, which is really cool. And why Antonella and I think they're so cool is because they're sort of changing the rules. So as Jouvay said, like, naked mole rats are the anti-mammal, right? They're changing everything that it is to be like mammal. But when they go through this disperser period, they're so much more like us than they are like naked mole rats. So they switch their physiology. They become completely different. And for a period of, we think, a couple of weeks, they become more human-like than mole rat-like. And I know that's got your mind going, right? But they don't, they don't start speaking and, and having tea. But
they drive a little car, and that's how they get to the.
Yeah, but no, the way that their world is, we could never survive in their world, and they have a hard time surviving in our world above ground. And so, they somehow flip the switch and can do perfectly fine in our world, which we couldn't do, right? It's not that we, if Antonella and I went to East Africa and decided to crawl into a mole rat burrow, we'd be passed out within a few minutes. So
that's not for lack of it's not for lack of wanting to, though, right, Dan?
That's right. Right. That's right. If they'll have us, yeah. If they're listening.
Yeah. And how deep are their burrows, and how does that relate to oxygen or lack thereof?
Yeah, great question. So Jennifer Jarvis started this whole idea of studying naked mole rats as a species, and then people like Rochelle Buffenstein and a bunch of great people. I'm blanking on names right now.
I'm sure it's not an Academy Awards. No one's going to
Yeah.
come out and be like, "How
That's
dare
right.
you?"
A guy named Brett went over and started doing measurements of mole rat burrows and found that generally they're about 6 feet deep, sometimes more shallow. So there's some parts of it, 6 feet, I guess, would be the maximum depth that you would expect.
Okay.
But they need to get fresh air. They require some. They can't live without any fresh air. So they do make these volcanoes, spaced out where they sort of break the surface. And what's interesting is that they have access to the surface, but they don't go there, except for these dispersers. So and you can tell that by the way that these volcanoes are formed, because you have this hole that goes above ground, right? And then this sort of circle of fresh dug material, right? And so the fresh dug material, if the mole rats were coming up above it, would be trampled on and you'd see paths and things. And you don't see that. So they just kick the dirt out, bring in fresh air, and move it around. But one of the important things for all eusocial species like bees, ants, termites, is the establishment of a nest, something that E.O. Wilson called durable and defensible. So something that's going to be around for a long time and is easy to defend. And those are sort of the key characteristics of a nest. In our colonies in New York, when we look at them, the first thing they do is establish a nest if we put them into a new environment. And we think the same is true for what they do in Africa, that they're going to establish a nest. And we think unless that nest is disturbed in some way, they're going to stay there. And so in the wild, you're going to have, 300 plus animals in some cases sort of piled on each other in this, this area the size of, I don't know, a bread box, I guess, if anybody knows what a bread box is.
We will be putting
That's
putting
how
up
small
pictures.
they are?
A microwave.
For that many?
Yeah, so they're just they're just piled on, right? So we did some measurements.
And they're like bigger than they're big.
They're big, yeah. Some of them are about
mouse
size, but most of them are larger. Yeah, they just stack on top of each other. And that's, so in our lab we do studies of that. What is the CO2 level? What's the air quality like in these environments? How many animals are in there? How much time do they spend in there? And they spend pretty much all day. They really love that environment. If we take that environment away from them, give them fresh air, they're really unhappy. If we give them the stale air that they like somewhere else, they're going to move towards it. So it's the most anti-mammal thing they do, is sort of like this really, we would cringe in that sort of environment. It's like we did the calculation once, and it's like putting 1,100 humans in an elevator, right? So it's just like, it sounds awful to everyone, right? But this is what
I'm not good with 10 people in an elevator, a large elevator.
Yeah, I was going to say, no.
Yeah.
Like, I prefer to ride solo.
Right.
Like,
and this is their day. They, if you take them out of it, they want to go right back and hang out there. And so that's what our research is really kind of, if you had to pin us down to one thing that we really are focused on, it's why. Why is the brain drawing them back to this environment that's so different? And we think we have some ideas. We've seen some things that seem to fit. And it's just an interesting, why would a species evolve to need to stay in this stale air unless fresh air is bad for them? And it sort of recreates this sort of desire for the animals to go back to their nest. And we think the system in the brain that's sort of utilizing this is about, things that we can relate to, anxiety, things like that, right? So we think, based on our observations so far and based on the systems that we know are kind of affected by this, that when a naked mole rat leaves its nest and goes somewhere else, unless it's doing work and staying really active, it starts to feel anxious. And that anxiety is reduced when it goes back to its nest environment, right? And so you're creating this reinforcer, right? You're creating this thing that's the mole rat says, I don't feel so great. Let me go back to my nest, right? And that creates a eusocial animal that never goes above ground and never has this desire to go and breed. And so here we are 39 million years later with the same species doing the same thing, right? So it's pretty cool.
That is fascinating.
And so listeners at home, I want to sort of paint the image of coming into these naked mole rat enclosures, seeing a bread box sized chamber full of naked mole rats, and having condensation on the lid because of how many animals are in there and how stale and sticky and humid it is that you would pop the lid off, they'd all scatter, and it would just peel off. Just running rivers of wet. That is how stale. That's the staleness that Dan is referring to. It's not just
they would love it here in Florida.
Yeah, they probably would.
It's crazy because it's like, it's like going to the International Space Station, right? And let's say that there's 10 employees working up there, 10 astronauts doing something, and somebody goes to visit them and says, let's go check out the space station. And they're nowhere to be found. And then you open this little broom closet and they're just piled up in the broom closet, right? They're not utilizing the space that they have. They're just they're crowded. They want this cramped space where they're all together. It's really amazing.
Mm-hmm. So what about fresh air do you think is bad for them or that they don't like? Like, is it too oxygenated or... Because I know even humans, if we get straight pure oxygen for very long, we have problems. Is that what it is for them, or do we know?
Do we know, Antonella? I'll throw this one to Antonella.
Oh, I was going to give it to you.
Hot potato.
Dan, you don't want it?
No.
Oh, no. Okay. So we think that they're using the high CO2 in the nest to suppress seizures, right? Or anxiety on a low level, right? Like hyperexcitability, So they have a genetic mutation that in humans, if we saw the same thing or when we do see something similar, the symptoms would be that of a seizure, right? Hyperexcitability. The neurons are overfiring, right? The brain is too active. Like when I'm speaking to you right now, even though we seem to think of ourselves like we're on, I actually have the gas and the brake on at the same time, right? That I'm not overfiring neurons. They're able to take breaks in between my words, in between what I'm saying, my thoughts, right? We have the brake and the gas on at all times. And so their brake system is what's mutated, right? That's the thing that's broken. And so we think that the high CO2 in the nest underground takes the place of that brake system, right? So that their gas pedal is not always on. So then they depend on it and then have to come back in. In the high oxygenated environment, they wouldn't have that brake system work as well.
Okay. Do you think then that this is like, again, all my mind blown. Is there the possibility that part of how they disperse, can they use the gas pedal overfiring to their benefit and like go really fast or swim or climb mountain? Like, you know what I mean? Like, is there a connection there where whenever they disperse for whatever reason, they can almost channel, instead of having a seizure, they just like overdrive to get to the next place?
It's like,
is that crazy?
No, Amber Lea, you're thinking of it in exactly the right way. I'm passing it to Dan, but you got it. Go ahead, Dan.
You're halfway to a PhD in our lab, Amber. So that's that was the critical halfway point that you just that's exactly the way we think. Like somehow these dispersers, they flip the script, right? So somehow, like we think, we don't, one of the hard things is that we are dealing with an experimental setup, right? That's, not quite what the mole rats had in mind, as they built their colonies and stuff. It's not like they were sitting there saying, oh, I wish these were plastic cages in Staten Island, New York, and not some. So we're giving them something that maybe isn't what they really, are used to. But we know that they survive and do perfectly well in our environment. They breed. They're quite content. We can see the same behaviors that are observed in the wild. So we know that they're living the life pretty much like they would without the threat of predators. And maybe, so there's some benefits to it as well, right? So we don't know exactly what the conditions are in East Africa. It's one of the things that we hope to one day learn. I talked to people who have been in Africa. I'm not going to name names because I might get in trouble if I divulge who told me this, but I have an anecdote from someone who went to East Africa and for whatever reason, took naked mole rats from their burrow, which they're catchable, that's how we got them here in the first place, and put them above ground and observed their behavior. And just like we would see in our laboratory, so if you take a naked mole rat out of our environment, put it in an environment that we simulate above ground, so it's 42 degrees Celsius or 105 degrees, so not the hottest day ever on the planet, but a hot day. Because naked mole rats lose CO2 just like we would as we start breathing faster, they can't handle it quite like we can. So they start, as Antonella said, they have a full-blown seizure. They have an epileptic seizure because they can't handle the hot, fresh air. It's just not what they're equipped for. If you take a disperser, and we've had a few, put them in the same condition, they just go to sleep. They're quite content. They're fine, right? And if you ask somebody who's done this over in the wild, they'll tell you that naked mole rats in the middle of the day on a hot day, if you take them and put them above ground, they'll flop over and essentially have a seizure and die. So we think what we're seeing is really analogous to what they would experience in the wild. So we think, we don't think that it doesn't make sense for animals to be, 5 animals a day venturing above ground and not making it more than 10 minutes, right? That doesn't make sense from an evolutionary standpoint. So we think that there is this important middle ground of, as the animal gets closer to fresh air, something kicks in that says, maybe we should go back to the nest. Something's off. I don't feel right, right? And so, as Antonella said, they're using the CO2 as this relaxation. CO2 is relaxing for us as well. So we've done yoga breathing for the past 4,000 years to relax, and we conserve CO2 when we do this, right? So it's no mystery that CO2 does something to our brains. We've changed the way we breathe and things like that to do that, to accomplish that. So the other thing that I think a lot about, and so CO2, Antonella was 100% right. CO2 is the answer, right? That's what we think is the key factor here. And the other thing that we like to think about is, well, what role does CO2 play in our life? I just told you that changing our breathing pattern is one thing, but the world around us is increasing CO2 every day, right? So climate change is affecting the CO2 that we breathe, and we are breathing more than double the level of CO2 that we did as a species when we first started. And projections are that it will more than double again in the next 100 years, right? And so that's going to have some impact on the way that our brain functions. What it did for the mole rats is it permitted these other things to kind of creep through, these mutations like Antonella's talking about, right? So if the air around me is relaxing my brain, then maybe the things that normally relax my brain don't need to be there. So it relaxes what we call selective pressure to keep the brake pedal on and keep the brake pedal working, right? I just got a new car, and it does this sort of thing that if there's a car in front of me, it's really cool. If there's a car in front of me, it'll start to slow down. So I use my brake pedal a lot less than I usually do, right? I don't have to step on my brake if somebody slows down in front of me. So it's exactly the same thing. But then when I get in my other car, I'm sort of not thinking about my brake as much, right? So then every once in a while, I'm like slamming on the brakes because I'm like, oh, wait, no, so my car's, so it's exactly the same thing, right? If the thing around me, if my environment is doing that braking for me, then I don't need to think about it as much. And it's the same thing that we think happened with the naked mole rats. So some point along their evolution, this high CO2 environment they encountered in their nest calmed their brains and relaxed the pressure for the endogenous calming, the inside calming to take place. And so things like this mutation broke through. For them, it wound up being a really good thing because then they can build up these large families, keep the CO2 going, and it wound up being a good selective strategy, right? So they've been around for a long time. They're considered pests in Africa. They're doing well because of that.
Oh, I didn't know that.
Yeah, because of that.
Whoa.
I don't think it would work so well for us. And so one of the things that we know, that the mutation that Antonella talked about in the naked mole rats, is that we know that humans have that mutation as well. And those humans are prone to more seizures. We see the mutation more in people with autism and schizophrenia. So we know that this mutation is capable of breaking through. And we would predict that with climate change, that we would see more of this occurring, that these mutations would be more likely to occur because the environment is helping us keep the brain calm with higher CO2, right? So part of what we try to think about is
I'm gonna break this in, because what is the mutation called?
Yes, it's you ready? This is where we sound really scientific. It's KCC2-R R952H. So There's the gene is
I'm gonna Google
it's
that.
the gene that encodes the potassium, which is a K, chloride, which is a C, cotransporter, which is the other C, and then the 2 just means which isoform it is. It's the one that we find on the neurons. And the R952H means that there should be something called an arginine in us and in every other species, every other mammal on the planet, except for one. Uh, Naked mole rats have an R there. And the other species that I talked about that is also eusocial, the Maryland mole rats, have something other than an H. But everybody else on the planet has an H there. They have a histidine. But these 2 species change that one spot. One has an arginine there, and one has a cysteine there. And we think that that might be important in the way that it works, that it's changing the way that it works in an important way. So that's kind of what we're trying to figure out, right? How important does that make these animals more environmentally sensitive? So it's funny, like when we think about mole rats, we think that they're sort of the word about them, if you know the literature, is that they're sort of environmentally tolerant, right? But we think they're sort of environmentally handcuffed, that they need to stay in these environments that they're in, and if they leave them, then they're in trouble, right? So that's part of what we're trying to figure out, is this KCC2 mutation in the brain, whether that is kind of leading the charge for that.
That's super interesting. And Dan, you know this, but other viewers might not. So my grandmother was diagnosed with schizophrenia. My family, it runs in our family that we're all dyslexic, which is associated with schizophrenia. And I wonder if I have done my DNA, if that gene is a little funky, just a little funky.
That would be interesting to know.
Yeah, it would be interesting to know. It would. That could be.
If I find it, I'll send it to you.
I have never considered dyslexia and KCC2. That's a cool. I don't think anyone has, Jouvay. I think you've just claimed it live.
Am I on the way to a
yeah.
to a PhD?
Yeah.
I was going to say, now you're halfway to your
there.
PhD.
Yeah.
Listen, I don't need any more. There's nope. Not doing it.
No
Dan
more
has
letters.
been trying. I am halfway to just sitting in my chair doing nothing. Thank you very much.
So this is a little bit of a tangent, but based on what you've just said about the mutation, do you know, and it's okay if you don't, because this is deep into the human realm, have people ever been treated for things like seizure or autism, or, the things that are associated with this mutation with CO2? Is there a CO2 therapy that happens?
Yeah, it's a really great question. So we were really fortunate to work with around 2019, 2020, maybe 2018. Also, the preeminent researcher in KCC2 in the world, who's a guy named Kai Kaila in Finland. And he took this, he was the one who really discovered, one of the key people who identified that this RH952 mutation in KCC2 had some importance, particularly around epilepsy. And they followed this family that has this mutation, and the family is in Sweden, I believe, and this mutation they have it in the likelihood of them having a particular type of seizure. So the seizure that they have, think about what we did to see a seizure in a naked mole rat. We put it above ground in a warm environment. So this is sort of a full circle thing, because the first thing that I wanted to do when I first learned that naked mole rats didn't control their bodily temperature was put them in a warm environment and see how activity in the brain changed, right? And then we ultimately got there. And when we got there, it was sort of so much more, astonishing, because we knew at that time that they had this mutation, that this mutation in the family that Kai Kaila studied in Sweden had a predisposition for seizures when they had fevers, particularly. So when they were warm and breathing faster and exhaling CO2, this is when these folks were having seizures. And so there's 1% of the population of children has a febrile seizure, a fever-induced seizure. And we know that there's genetic predispositions, so that, you have 9 kids that have a fever, one that has a mutation in KCC2 may have a higher likelihood of having a full-blown seizure in response to that fever. So it turns out that those are the people that have the same mutation that the naked mole rats have. So what has Kyla done? In Finland, in Helsinki, where he works, they have 5% CO2 bottles in every emergency room. Anybody shows up with these fever-induced seizures, they do an inhalation of 5% CO2, and it just as he showed in the mice and we showed in the mole rats, that aborts the seizure, blocks the seizure, and prevents it from happening. So,
oh my God.
So the answer is yes. So people use CO2 to treat seizures, and in fact, Kai's postdoc, Martin Puskarov, who came and did some work with us, he sent me this really cool picture, which is from the 1500s, a painting by Grunwald, where they showed that the physician to the king, I don't remember what country it was, but the physician to the king was strangling a young child, had a sort of this scarf around their neck to treat the child's seizure. So he was cutting off air supply, increasing CO2. So, we think, Kai Kyla, how brilliant, 2008, 2009, when he started doing this. I mean, I think we learn these things over and over again every few hundred years, right? These new treatments, right? So people have been using that treatment before, and then modern medicine came along and said, let's do away with that CO2 stuff. Come on, no strangulation. That's crazy. But it turns out that that was an effective approach, right? So I'm not advocating for strangling anybody, just to be clear. But it was something that was done, and but
I have to wonder the first time was
That's
somebody
exactly
just like, this kid's driving me crazy. Like,
that's right.
Like how did they discover it?
Yeah, it's true. That's true.
That is fascinating. Wow.
I didn't even know that. This is all new for me too.
Oh, that's exciting.
Good.
Yeah. See, I don't know everything that there is to know.
Okay, fine.
Thank you. I appreciate that.
That's amazing. This is so cool. And so I think let's piggyback off of this application of mole rat to human similarities. And can we talk about how they don't get cancer or are very resistant to cancer? Because I think that's a thing. There's never not going to be an interest in cancer research until it's eradicated. And when Jouvay first told me that they just kind of don't get cancer, I was like, how are they not a more popular animal? Like, shouldn't they be on like stamps? And like, this seems very important.
Yeah, it's funny. I have a colleague who's a cancer researcher, and I said, hey, would you, we have some tissue from naked mole rats. Are you interested in having it? And he said, I'm a cancer researcher. Why would I want somebody that's already fixed cancer? So their job is to make cancer so they can try to, you know what I mean? So it's like, I didn't think about that, but I'm like, you know what, you're right. They've already solved the problem, right? But there is a really great researcher named Vera Gorbanova at Rochester who had this really important paper, I think 2013, and she showed something that prevented cancer cells from sort of connecting with one another. So if you talk to cancer researchers, one of the things that I learned, and I'm not a cancer researcher, so I don't know much about it, but one of the things I learned from cancer folks is that in some ways we're all kind of, and this is a scary part, right? But in some ways we're all walking around with sort of cancer cells in our bodies, right? That are mutated, right. Yeah, the cells that aren't doing what they're supposed to be doing. And the cases where cancer develops, you just have this aggregation of these cells, and then they start reproducing and going haywire. And so I think what happens in the mole rat is that, based on Gorbanova's work, is that these cells don't have a way to connect up. And she showed in that paper that there's something really interesting about a key molecule in their skin and really all their tissue, their brain and everywhere else. And it's a molecule that if you have watched a commercial for cosmetics in the past, 4 years or so, you've heard of, which is hyaluronic acid. So hyaluronic acid is this thing, once you hear it, you'll be looking for it in every commercial now. But watch a commercial with shampoo or with moisturizer, and they're talking about hyaluronic acid. And what hyaluronic acid does is it holds a lot of water. It's really good at holding many molecules of water. And naked mole rats have a very special hyaluronic acid that holds even more water than most. And we think that that's important for a couple of reasons. One, they live in the desert and they don't really get a lot of water, so they have a good mechanism to conserve it when they get access to it through the vegetables and things that they eat. But also, it probably keeps their skin soft as they move through their underground burrows and prevents them from getting tears in their skin and that sort of thing. But
So
yeah, they are. They're
they
really soft. They're really soft. Like I'm a believer in hyaluronic acid. You know, like if I see the commercial, I'm like, yeah, get that, because I know how soft these animals are. They really are so soft.
That's so funny.
Yeah,
And
so,
they feel nothing like how they don't feel how they look.
yeah,
They feel
they
feel like,
I was gonna say they look like, I don't know what I thought I think I thought they would this is why we're no filter. I think I thought they would feel like old people. Like
yeah,
that paper.
yeah, yes,
no,
right,
they feel
no, they're just wrinkly babies.
yeah,
They're
yeah. It sounds like they feel more like
yeah,
babies.
like
Yeah.
suede.
I wonder. I'm gonna have to like go down the rabbit hole on this. If there's an association with hyaluronic acid and skin cancer, like if people who use it regularly get less skin cancer, or if that has nothing to do with it. I have no idea, but I'm curious now.
That's a great question.
Because that would be kind of the application that we could see in humans as the most because it's the most common way we use it.
Yeah.
And it would be interesting to know if that could be a mechanism that translates to humans and isn't just a special mole rat thing.
That sounds like a second PhD right there.
She's on her way to her second.
We're just racking them up, Amber Lea.
I'm really good at coming up with research ideas. I'm less good at like executing.
I am the same. I'm the same.
I think this is a room full of that right now.
So I want to, you mentioned what they eat. Can we talk about what they eat and where they find it and how they get what water they do get? And also, this is also just a kind of a, let's segue also into their teeth, because I love their teeth and I would like to talk about their teeth, but also their diet.
This is where Jouvay's expertise. I'm throwing it to you,
All
Jouvay.
right. Yeah, Jouvay, why don't you tell us, while you're answering my many questions, a little bit about your master's project, because I know it has to do with this.
Oh, goodness. It was indeed a Home Depot project that got me a master's degree. And so way back in, when did you try and start, Dan, with trying to fix their enclosure?
It was pre-COVID for sure. It's 2017, 2016 maybe. I don't know.
Somewhere about there, you were trying to. Right now, the enclosure that is there, that our guys, is a very modular, really cool setup, but it can be a bit of a headache because it's a lot of twisting action. And so the techs that are constantly working that enclosure, it wears on their wrists. And so Dan, being a lovely human, saw this and wanted to change that. And then also, I feel like there were other research reasons that you really wanted to be able to change that. And you had given that project, I remember, to a couple of undergrads who kind of ghosted you.
Which happens.
And then
not unusual.
Yeah. But for a very good reason, because as I started to do research, I started to realize how involved it was. And so basically what we learn when now there's two mole rat Bibles, right?
Mm-hmm.
There was just one.
Yep.
Back in my day, there was a mole rat Bible, and one of the first things that you learn from Brett, that's his last name or his first
Brett,
name?
last name.
Brett, last name. Scientist Brett, thank you, Dr. Brett. One of the things that you learn from the opening pages is that these guys basically can chew through like solid rock. Like they're living in like pure gypsum, and they're able to chew through it. And when you start looking at husbandry manuals, they all basically say the same thing. Check on your animal enclosures like every 6 hours for holes. Right? So these guys are constantly using their teeth. And so a rodent is a rodent because it has two sets of ever-growing incisors. So these incisors throughout their lives will grow a couple of fractions of millimeters throughout the day. And they are super hard. They are super durable. They're super good at chewing through pretty much anything. I mean, rats can chew through cement. Um, and these guys are more adapted for digging life. So much so that their teeth erupt in front of their lips. So their lips kind of form like a little seal, and their teeth are
This is
out
the
here.
thing. This is the thing that I just need people to know. Because it is insane.
It is insane. But there are other animals that have this like teeth in places they shouldn't be, right?
Who else?
Narwhals being a great example.
I was thinking other rodents. I'm like, tell me now.
No. Warthogs are also another really great example that their teeth erupt in their cheeks, essentially, through their skulls. So it's not necessarily an unusual thing, but for them, it just adds to the funkiness. And so these guys are able to chew through this environment. And Dan mentioned the relaxation of selective pressures. And one of the jobs of any husbandry person is to essentially maintain those selective pressures in some way, whether it's in the zoo field for preserving that species to be reintroduced, or it's in the laboratory field to make sure that those behaviors and the things that make that animal studyable are preserved, right? Because if you start to somehow relax them enough, they stop being those naked mole rats that may love that CO2 as much, that may, have that physiological weirdness about them that makes them such a icon. Really, they're icons. And so I, coming from that husbandry scene, wanted to make sure that we found a way to keep those selective pressures, to keep up, to give them the context that they evolved in over those millions of years. So we started with Portland cement, and then the constraints of laboratory life were, well, it can't be that heavy. So we added paper to it. The paper and Portland cement were super dusty, and we needed to have an easier way to clean up. And it just so happened that I just basically took a bunch of paper pulp, made it paper mache, and then smushed it and let it dry, and it turned into a really solid block of something that I couldn't even punch through. And so
and just so everyone knows, Jouvay does do karate, and so that's saying like that's it's right. Like not to be silly about it, but really, like,
yeah, no, I was trying. I was really trying to punch through it, and I was hurting myself as someone who I could do push-ups on bare wood floors. It hurt. And so from there, what did we put it in we made a giant column of it, hooked it up to one of the colonies, and overnight they had made this little spiral staircase in it. They chewed straight through it, and we'll put some pictures up in post. But they chewed straight through it, and they went up to the top, and they just freaking loved it. And that was the first time we were able to give them 3 dimensions, right? It wasn't just a flat enclosure. This was them choosing height too, and that's important because, as Dan said, their whole entire colony spans 6 feet at least and miles wide. And so we weren't giving them that, and we think that it's, the height is important for thermoregulation too. And so that was another thing that we were able to give them and afford them that they hadn't had the opportunity to. And so we've now kind of looked at risk assessment of, is this a safe material? And we're still in the process of looking at the data, but we've back to that publishing and how long it takes. But what we found was it's fairly safe for them to be in a recycled newspaper pulp environment and to climb up in it. And microbiome wise, it's also a really interesting thing that's at play. Like, there's whole ecosystems that are happening around them too, which is really fun. So that's their environment and in the lab, and we supplement their diet in the lab with sweet potatoes, any sort of tuber, so any water storage body of veggie, In the wild, they are a nuisance because they will go through sweet potato, cassava, and yucca farms, and they will dig out the fruiting of these tubers, and they'll leave the shell though, and they'll pack it with dirt so that way they can come back to it so it regrows. So they're farming. They're farming little cute naked little sausages.
This is
Yeah.
Nobody is reacting. That is so cool. That is, and you're not reacting because you all knew this already.
Yes. Yeah. It's old news for us.
I am so fascinated by wildlife that cultivates and farms. So one of my favorite things is dugongs farm seagrass, and the way they eat seagrass causes like gardens of seagrass to grow. I love that the wild mole rats have figured out how to utilize what humans have done in the earth and then build upon it.
One of the driving factors of their evolution is thought to be the sparsity of food. If you're in a desert, your food is in patches. And so, maybe my thought is they're probably doing the same thing that elephants do, is navigating to these places that they know are good soil conditions where certain tubers or even succulents will grow that they can munch on and then come back for later. And I think what's funny is that all of the food that's grown in Africa right now, in East Africa, that they're a nuisance on are non-native. So sweet potatoes are non-native, yucca non-native, cassava is non-native. So these are all things, and these are all the things that we feed them in the lab. But when I, as a side quest, went to get a tattoo after I defended my thesis, I was like, I'm going to get a tattoo to get over the pain of grad school. Although Dan made it very easy, let me tell you.
There's always pain. Always pain.
There's always pain in grad school. And so to get over that and to celebrate, I went to go through even more pain. But I got the naked.
There's a lot of endorphins involved in getting.
There is so many endorphins, so many good endorphins. The tattoo.
Have I seen this tattoo?
You have to show the design in post production. It's awesome. Yeah. It is so cool. Beautiful.
So when I got the tattoo, it's of a naked mole rat and a Damaraland mole rat, because I did take care of both of them. And I wanted to find their native plant. And when I tell you, I spent hours and hours down a rabbit hole trying to find a native tuber. It is difficult because they're not, there's so little information on what they eat in the wild that isn't in farms.
Wow.
Yeah. So I had to choose, I think it was like an arrowroot, which was a non-native thing that they eat. I will say, Dan, Antonella, if you guys need to go at any point, you're more than welcome to. I would love.
Don't echo that sentiment. I have 1,000 more questions.
I would love to give Antonella the opportunity to talk about her PhD work, though.
Yes,
because I would love to hear about that. You don't have to. It doesn't have to be sparkly. I'm not. No dissertation defense.
Right. There's none prepared, so we're on the same page. Yeah.
So is your PhD work separate from the dog stuff that you're? Oh, okay. I thought that was what you were focusing on. This is cool. So
yeah. I mean, unless it just turns out that way later, right? But for now, but as I learn about the mole rats, my wheels turn about the connection with the dogs. That's what happened, right? And humans also. But so I'm gonna answer Jouvay's question. If I can. So for right now, I look at the actual nest, Amber Lea, and how they build the nest, right? So perhaps we know why they like their nest, right? Everything we've spoken about, right? That brake system, the CO2 that they need. But when I started, I was just watching hours and hours and hours of mole rat videos, just staring at them. It became my favorite TV show. I was just watching them live on the camera. And I've been doing that since childhood. Any animal that I ever rescued and brought into my home, I would not go to family events and just stay home and watch my birds eat, watch my dogs drink. How do they do that differently from a cat? What's the difference in the shape of the tongue, the action of the tongue? How do these animals resolve conflict, right? And if I'm living in a multi-species household, how do they resolve conflict in captivity, right? All of these things that I've been doing from childhood, I brought now into the lab because I have no choice, right? That's just the way my brain works. So I was watching these videos, and one day it just hit me that I was seeing this repetitive behavior that, like I said, the different groups, right, the subcasts of mole rats within one colony were doing seemingly predictably, and like they liked it, right? And so I showed Dan, and he let me go for it, right? And I just dove into this, how they build the nest, right? How different subcasts of mole rat build their nest. So that's the path that I'm on. I focus primarily, I mean, there might be, other ones that come up later, but the two main subcasts, there are small workers and large workers, and they seem to have different requirements to initiate the nest, join the nest, build the nest, maintain the nest, right? How they do it and why they do it might be motivated by different things. Physiologically, socially, might be motivated by different things. So that's the deep, that's the rabbit hole that I'm in right now.
I'm excited to see what comes out of your research, because I think that is a really fun topic that, of course, also could be really important to, further study and their conservation and, insight into kind of how they live their lives.
So am I, but one of the problematic consequences of that is if I was, like, let's just say, for example, which I hope that you do, come in and see the naked mole rats, right, in Staten Island.
Oh, what I must touch a mole rat.
when we walk into the room, typically what happens is I have to pause and stare at the nest that I find for as long as they're willing to maintain it for me, because to me, it's so beautiful to see, right? And to other people, initially it might seem like they're laying around sort of like haphazardly on top of each other. But when I look at that, there's the preliminary, steps that they took to achieve that, right? And it's fascinating.
Mm-hmm. So is there an order of who gets to be where?
I think so.
Like, is there like this is the better spot to lie?
I think so. Okay. You ask me in a year and I'll have a
okay.
I think so. Yep.
So I'm one of those people that Antonella talks about that sees haphazard, right? That's working with these animals probably for 10 years before I was lucky enough to have Antonella join the lab, and looking at a nest and say, yep, there they are again. There they are again. Yep, there they are again. I knew it was important for them, but to her credit, she sat and really, really picked it apart and looked at it. And when she first said, there's some pattern to how they form, I think I didn't believe her. Right? Antonella. And then she would show me video after video after video of the same sequence. And I'm like, oh my God, it's real. It's real. And so that's so it in a way, it kind of, in some ways, makes what I said before a lot more complex, right? So what I said before was they're getting something out of the nest, they're returning to the nest, right? So that's like the reinforcer. It's something that draws them back rather than go run away, start their own colony. They return to the nest because it feels good, right? But what Antonella's stuff is showing is that it feels good for different reasons. Otherwise, they would all do the same thing, right? So if everybody got the same thing out of the nest, you would expect a very haphazard, who goes where, it doesn't matter, let's just nest guys. But that's not what she's seeing. She's seeing a specific sequence of behavior, different animals doing different things, and that's showing us that they're probably getting different things out of it. Which, if we bring this back to the humans again, right, maybe there's more than one way to treat people who are having things where their GABA system, their KCC2 isn't working right. Maybe for some people it's one thing in the nest. So treating the nest as like this almost like this organism, which is, really the way Antonella's approached it, and figuring out what makes the organism comfortable and why is this part doing this thing and this part doing this thing might open the door for us to see like different possibilities, which is really cool.
One important thing that I have to mention is that we challenge them to nest in situations that would otherwise be very uncomfortable for them. And they still, if they don't fully accomplish it the way that they would in their natural setting, or at least their home setting in the lab, they at least attempt to, and they will keep trying until they get it, right? So it must be very important to them, because if you put me in an uncomfortable situation, the first thing I'm not going to do is look to sleep, right? Or look to relax. That's not the first thing I'm going to do. It'll be on my list somewhere eventually, right? But it seems to be a priority to them. So that's something, so it jumped out to me as something important. And I think that I still see those same patterns between the subcasts, even in the uncomfortable environment that they're not used to, with typical cues that they might have to nest in the home that they're used to.
That is fascinating. It sounds more complex and complicated maybe in the nest scenario, but are there other scenarios where there are role specializations? I see Dan nodding.
Dan,
Antonella is too. I know she knows this because she's working on the same paper I am. But we developed, so one of the things, when you have, some of our colonies now are 100 plus animals, and
oh wow,
yeah, so it's one of the things that becomes difficult is to figure out who's who, right? So what we did early on was put little RFID chips under the skin of every animal, and the RFID is easy pass, same technology we have in our cars to track our tolls and charge us money. We don't charge the mole rats, but we can see all of their movements. We see every place they go, where they are, and all this stuff. And we realized, first of all, it's a really cool way to track what they're doing. But second of all, we can use that to make some fun tests for them. Here's our sort of problem, right? So traditional testing in my world, in behavioral neuroscience and psychology world, is taking a rat or a mouse, putting them in a new environment by themselves, often in very open environment, and then running some sort of analysis, right? But we have animals that really shouldn't be separated from each other. We have animals that really detest fresh air, and they're not used to new environments. So if we do any of those things, we're not going to get true behavior, right? So we figured that we should try to figure out behavior where we can sort of measure what they're doing in their natural, semi-natural habitat, right? And we definitely do see different roles. So what's interesting is that in the literature, the early literature from the 90s and so, Antonella said that there's small workers and large workers. And large workers got the unfortunate name in the 90s of lazy workers. So the early studies suggested that the large workers do nothing but sit around all day, except when there's like a threat. So somebody, I forget who, put a snake in the cage and saw that the large workers went and attacked the snake. And, we don't do those kinds of studies today, but those were the early studies. But the large workers clearly had a role in defending the colony, right? But a lot of those studies were done with the sort of problems that I talked to you about, that they're taking animals out, putting them in this weird environment and seeing what they do, right? And so they found in those situations, the small workers did more of the things that they tasked them with. But when we look at it through our Easy Pass and, the RFID chips, we see that the big guys do most of the work. They do almost everything there. But the small guys play a role when we put a foraging type of environment, when we add something on, and what we do is this sort of big thing full of sand that they need to endlessly dig through, and the small guys seem to gravitate towards that and dig through the sand, and the big guys stay back by the nest. And so we think there's sort of this zone system that they create where large animals, because protection is their primary role, I believe, because that's the only behavior where we really see 100% large animal, if you create a threat. And so our threat is just to create the sound and scent of a foreign mole rat. So, record a different mole rat from a different colony, making this alarm sound, and then take the bedding from that colony and put it in, and just put it behind a barrier so nobody can access it, and that will recruit the large guys to come right away and try to defend the colony, right? So 100% that is their behavior. That's they own that. But pretty much everything else everybody does. But as you get close to the nest, if you have a small animal bringing a piece of cardboard to the nest, you'll see a large animal rip it out of its mouth and then take over. So they create this, anything near the nest, it seems like large workers say, this is my territory, let me do this. And the small workers, we think, are doing stuff outside. So they definitely do develop roles. And we are seeing some changes in the neurochemistry of the brain that kind of accounts for that, right? So, because I told you before, if you're away from the nest for a long period of time, you might start to feel anxious, but we're seeing that maybe the small workers turn on a chemical in the brain that might help them to overcome that, which again, maybe something we can think about for humans that might be beneficial.
Yes, please.
I could really use that because I don't like to go to the grocery store.
Yeah.
Yeah, for real.
I think if I had a chemical I could turn on when I go forage, that would be really
exactly.
helpful.
Right.
And so
that is fantastic.
Before we let y'all go, because I know Amber Lea won't let you go. But I will let you go.
I sure won't.
I would love to touch on how you were using the second species that we mentioned, the Demaralans, to aid in your research. This is, for listeners, this is mainly selfish because I was their primary keeper, and they're my favorite little toothy devils. So they are vicious.
They are vicious. Yes.
Compared to the nakeds, although our nakeds were said to be the nicest.
Oh, that's sweet.
From what we're told. Yeah, other places said that the people would get bit up. Ours, you could hold them like little rice croquettes, and they're just lovely dumplings, lovely little dumplings. The Damaras, on the other hand, quite smoochable only once.
Right.
Yeah.
Right.
But if you saw them side by side, size being the Mars are a lot bigger than the naked. So if you saw them side by side and you said pick up one, I would go for the furry one, I think. But the furry one, the Damara would take my finger off probably. So Jouvay is my hero for all the working with Damaras. Antonella and I are still trying to fill in Jouvay's footsteps and getting the courage up to work with the Damaras more. But so one of the nice things about doing naked mole rat research is it's a really nice community of people, researchers who are involved. And Shelley Buffenstein, who is, if Jennifer Jarvis is the queen or the initial colony, then Shelley Buffenstein is sort of the new iteration of the queen too. And, brought the animals to the US, did a lot of really wonderful research. And the reason we know about aging is really because of her research. And she's done, and cancer resistant, done so many important studies with naked mole rats. So one day she reached out and said, I have these Damaras and I'm wondering if you want any. And I said, yes, please. This is a great opportunity. We knew at that time that they did have this slight modification in the KCC2 gene that's different from all of us and every other species, right? So naked mole rats are unique because of one single point variant, and Damaras are unique because of a separate single point variant in the same exact place, right? So if for nothing else, we can start to try to tease apart what that difference might make. And, genetically, Damaras and naked mole rats are very, very similar. They're the closest living relatives. And as I said before, they're the only two eusocial mammals that we know of. So it's really, we've got the market, right? We're able to study every single eusocial mammal on the planet, right? By walking in 2 rooms. So it's really cool. So it turns out, and we don't know yet whether or not it's because of this switching in R to a C, in this one place on the gene, but Damaras are very, very different in many, many ways. So take everything we talked about, the piling on in the elevator, the seizure in a high temperature, the we don't know much about their social roles yet, about how they do that, but they're even shorter lived. They're much bigger. They're furry. So there's so many things that are different between these two species. But for the stuff that we are focused on and think about, we don't think that they need the stuffy air quite as much as the nakeds do. We don't think they're handcuffed to the environment quite as much, right? And we definitely know that they're not as seizure prone as the nakeds are. So they become this sort of perfect counter for us to see, okay, well, we can take the nakeds and try to give them a little bit what they have in the Damara, try to switch this one thing, change this thing, and see if we can kind of fix a lot of the problems that we see in nakeds in normal air. The other thing that we think, based on what we've seen, so Damaras will definitely have a nest. Antonella has been working with a really great high school student, Rachel Levin, who's been collecting air in the Damara colonies and the naked colonies. And as we suspected, the Damaras don't have quite as high CO2. They don't seem to need it. But we see that there's differences when they're out of their nest, that maybe they're actually generating more CO2. It's really interesting data that's coming in. But yeah, so they're really, we've just sort of hitting the tip of the iceberg with what we're studying with them. They're really interesting, and they're even more rare than the nakeds. There's only a couple of labs that are studying, so we have some really cool questions about differences in nesting behavior, differences in physiological need, differences in social structure, and all of this. We think maybe if I had to guess, I would guess in the wild, Damaras would be much more comfortable slipping in and out of eusociality than naked mole rats. They're not handcuffed to eusociality. I think they're doing it as a convenience, but I think they don't need to do it, which is really cool. Do you want to add anything, Antonella?
No, I was going to say the same thing. They're like facultatively social or something, right?
Yeah,
they're not obligated to be social.
Yeah.
I mean, that would be my guess, because when you look at them, even in their cages, they're out of the nest working and making noise so much of the time.
Oh, wow.
Even when no one's in the room and it's a quiet room, you hear them making noise and trying to dig and burrow, whereas the nakeds, they're looking for a quiet spot and they want to nest most of the time. So,
so it's much more like our what you'd expect for our space station, right? People doing work all over, spread out. Everybody got their space. So they're, they're definitely closer to humans physiologically than nakeds are, for sure. So it's cool to see that, we see the nakeds sort of this extreme, Damara's this halfway point, and then the rest of us, which are sort of on the other end of the extreme. And so the Damaras are this really cool, evolutionary, interesting sort of inflection point where you could see them going naked direction or the rest of us direction.
Wow. And where are they native to?
Same areas. Yeah. Same.
Okay. Yeah. So there's not an environmental difference that would have brought.
It's slightly different, right, Jouvay?
Necessarily.
Yeah. They're a little bit. Oh god, the sun is like making my arm white.
You're glowing.
I'm glowing. So whereas you'll see on the range, like nakeds are hugging the coast, these guys are a little bit more inward.
Okay.
Which probably makes more sense because they're usually dealing with more temperate climates. They're like at the top of South Africa and stuff. Mm-hmm. And I believe they can go up to higher elevations.
Okay.
Yeah, I think that's right.
So they're a little bit more, they're environmentally tolerant. I also happen to think that they're patriarchal instead of matriarchal, like the nakeds are. Because just anecdotally watching them, the males of each colony would be bullies. Whereas it's thought that the female queens in the nakeds are the ones that are kind of doing that bullying or suppression of roles. And also in Damaras, so the queen in the nakeds is usually the biggest, and you can usually point it very easily. You can't do that as easily in the Damaras. The queen isn't the biggest one by any stretch.
Okay. I can talk for an hour about what I'm about to say. I won't do that to you guys. I'm obsessed with bonobos versus chimpanzees, and how the theory that bonobos evolved to be matriarchal and peaceful because they had access to resources, and chimpanzees are patriarchal and very violent. And it, like, Jouvay, so much of what you just said mirrors that.
Mm-hmm.
And I'm like, fourth PhD.
That's right.
We study the evolution of the two species of mole rats as it relates to resource abundance.
Yeah, and you see, I mean, the matrix, just like bonobos. So bonobos and chimps, there's a lot of sort of hierarchical infighting within colonies. And if you walk into a Demara room, you'll just hear these little fights breaking out all day long. Whereas the nakeds, it's
trying to work, and they would be so distracting because they're yelling.
They just.
Just screaming at
Jouvay is right. If you walk into at least our Demara room, and you go to the cage, it is the breeding male who is right there, ready to get you. But if you go into the naked mole rat room, well, most of them are going to scurry away. They're just more timid to begin with, right? But I think that if go ahead, Dan. Yeah.
I was thinking the same. The queen is the last one that you'll find, for sure.
Yeah.
The queen is good at getting away and staying away. Yeah.
So that works for them. Whereas for probably the Damaras, I guess this other way. I mean, he's big and he's really scary looking. So I'm sure it has worked. Yeah.
I'm picturing like the Rat King from the Nutcracker.
Honestly, so like, whereas I would get like reverse trauma because the days that I would go work in the nakeds, I would open the thing and wouldn't have to like fight. It's like a New Yorker like moving to like the country or something. Where everyone's like, "Good morning," and you're like, "What kind of thing?" Like that's what it felt like because the nakeds are so much more peaceful, and the Damaras, as soon as you open up the lid, they're at you. And they are ready. They're ready to fight, teeth bared, and they have their incisors erupt before their lips. So you just got these big incisors staring at you.
Ooh,
and ready to go.
I'll use a dog analogy. I hope that it makes sense to you guys, but picture like an Akita or a husky that just lays there and waits for you to come near him. Look at me, come pet me, I'm so furry and cute. And then he'll get you, right? With almost no warning. That's what the Damaras seem to do. They're so cute and they come toward you, but they have these big chompers in the front that can probably take your finger, right? I mean, I don't want to give it a try, but I'm sure they could.
Yeah.
No, they went straight through my finger once. Like straight through.
Oh my God.
And that's on good handling, kids. I want to thank the both of you so much.
Thank you
for
so
coming
much
on and doing this with us. It has been lovely to see you both again.
Yes.
Likewise.
As it has been a long time. And I promise once Amber Lea comes back to New York, we will both take a trip over. So you
100%.
can meet, all of these little toothy babes, the saber-toothed sausages, as they are referred to in some articles. I won't say the other name because we'll get PG.
We can imagine.
Yes, but thank you guys so much. So very
Thank you so much. It was so nice to meet both of you and get to pick your brains. I really appreciate you taking the time.
It's a fun conversation. Thanks for the invitation.
And to our listeners.
Yeah, thank you guys.
Yeah, absolutely.
Posted.
Holy fucking shit. I knew this would be cool. This has so far surpassed my expectations.
Yeah. Yep.
I could literally talk to them for 2 more hours at least.
I told you there was a higher power than I.
I'm so excited.
And it was not imposter syndrome. It is because there's so much more about them that I could not even touch on.
I feel so lucky to now be part of, to have an in with the Naked Mole Rat community.
In. Yeah, yep.
Which I already did from you, but now I feel like I have an even more in of, I have formally been invited to come pet a mole rat.
Yeah, it's an official thing. They're, for listeners, I wasn't going to say this in front of them, but they're saber-toothed scrotums is what they're called.
So when we were talking about what I thought they would feel like, I did think they would feel like old people. I also thought they might feel like a dick.
That's mainly what people think. But they don't. They do not.
No.
They feel like
I love this.
They feel like
so cute.
Yeah.
So my two final questions. I know you have to go. So one question is for me. One question is for my friends. Question for me, super simple, super easy. I think. Do they fart?
Oh my God. Okay,
or do we know?
So I know that the naked. So one of the interesting things, and one of the cruxes of why my thesis could work, was because they have very similar gut systems to ruminants. So think cow. So they have a modified gut system that basically functions as a ruminant digestive tract. And so we were able to find papers where farmers were using recycled newspaper as feed filler, and see how it reacted to their animals. And so we were able to say, well, there's precedence for this, and we can move forward with it. So I'm gonna assume that they fart because they have a modified gut. I have never heard a mole rat fart. Second question.
Thank you. Second question. You had told me something, and I mentioned it to my friends, and then I couldn't remember whatever. I feel like you told me something about how much each mole rat is worth, and I feel like you said something about they're like very expensive. Did I make this up?
No, but you are confusing two different species. So it was a mouse model that was super expensive. So for those that don't know, if you have a transgenic animal, it can cost anywhere from a couple hundred to a couple of thousand to tens of thousands to create that animal, or to even breed that animal.
That's what you were talking about.
Yes, so each one can be worth. But, to keep a mole rat, you have to not only you can't keep one mole rat.
Sure.
The smallest colony that my former institution had when I was there was about 30. The largest was over 100.
Mm-hmm.
In the literature, they can get up to hundreds of animals. So, and they're breeding constantly, right? So you can't, unless you give them birth control, you can't stop them.
Which was a follow-up of the question is, are they all just bred now? There seems to be no problem breeding them.
You can't stop them.
Great.
You can't stop them. I mean, there are, zoos do use birth control and stuff, but, you really... It's not something that could be stopped.
And then there isn't a need to bring in additional members from the wild, since they're having a great old time.
Yeah, they're just having a slam bang time. So because their whole genetic shtick is being inbred, the woes of genetic inbreeding depression aren't happening. If they are happening, they're not happening as quickly as in other species. So we're not having to import mole rats to sustain genetic diversity or anything, which is
because it's no different in the lab setting than it would be in the wild.
Right, exactly.
Cool.
So with that and breeding, it's kind of just you set it and forget it, and they kind of do their own thing.
I love that. How big are the babies? Like little tiny fingers?
They're tiny little babies, tiny little, maybe to a knuckle, an inch or so.
I love that.
The Damaras, on the other hand, come out maybe 2 inches long and full furred, and they're big. So Damaras are the size of a big sweet potato, whereas your naked is, I don't even know, way smaller. So
are the nakeds more like a shallot, perhaps?
maybe.
I'm trying to think of something that's small but not totally round.
Like those little oblong potatoes. You know those little guys?
Oh yeah, like the fingerling potatoes.
A fingerling potato. There you go.
Okay.
And with that, we are going to close up our second episode of No Filter Fauna.
This has been fantastic. I have learned so much.
Yeah. So,
thank you everybody for listening and watching, if that is what you did to consume this podcast.
And we'll see you next time.
Yeah, we don't know when it is, and it'll be a surprise for all of us.